Now, the very few people in SSE would be able to listen to these takes. One of them is Dr. Mohammed Saeed, professor in chemistry. Without further ado, I would like to take the stage and walk us through this brilliant journey.
Assalamualaikum. Ladies and gentlemen, thank you very much for showing up for this presentation. So I just want to describe a little bit chemistry and a lot of general ideas which led to this Nobel Prize. So we will start with the very basics. And actually, I just want to admit at the beginning that I have tried my best to avoid the technical jargons and the things which are not obvious to many people from other areas. But somewhere if you see these technical details, then I think you simply forgive me for that. Okay, so what is chemistry and what chemists do? So if I ask you this question, then what comes in your mind? Well, I asked the same question from my about chemistry in our part of the world. So for me, if you ask me what chemistry is, I will say that to make this beautiful building out of these building materials or the building blocks, without even able to see them and touch them. And all these things are possible only if you apply the physical laws in an innovative and intelligent way. So chemists normally make molecules by using all physical laws and their applications. And then second question comes in mind that why we really need to make these molecules. What is rational behind it? When nature is making all sort of different molecules, then what is need for a chemist to do this job? Well, I can simply show you some areas where we really need to make these molecules. First one, this is a very nice, beautiful tree. It's a yew tree, and it produces a big molecule which we call paclitaxel, and that is breast cancer medicine. And if you cut a 100-year-old tree and dry the bark of that one, then you will get a couple of gram of this compound from it. And that is kind of time and the amount which we will get if you want to isolate paclitaxel from the natural sources. So you have to synthesize that. You have to find a way to make this molecule. Plus, there are many molecules which either nature does not like to make, or it is not evolved to that level. So we have to innovate some molecules so that they can become interesting medicines. In area of biology, molecules are designed to intervene or investigate different biochemical pathways. So right now, biochemistry and molecular biology, that is developed because of making a lot of molecules which are continuously utilized in this science. And for for sustainability of life, we have to make molecules as fertilizers. We have to make molecules as insecticides, pesticides, and not only to grow the crops, but also to increase the shelf life of different produce. So all these things require the synthesis of molecules. Now, when you are ready to make molecules, then there are different challenges. The first one is that most of the time the preparation to make molecules is a very lengthy process. It is a multi-step reaction, and sometimes it produces byproducts and waste material which are not eco-friendly and environmental friendly. Longer times and low yield, high cost. So all these are challenges when you prepare molecules. And in 1990, before getting the first Nobel Prize, very Sharpless actually make a comment that we have to modify our strategy to make molecules. We have to stop imitating the natural process, and we have to invent some reactions which he called the click reaction. So from there, this term was introduced for the first time to discover reactions which are modular in nature, meaning that which can snap together, which can behave like Legos. If you have, these, Legos which normally kids play with. So you can build many interesting articles if you have the Legos. So the reaction should behave like that. That you take molecules, they should snap into each other. They should be easy to carry out. Just like a kid play with Legos, then chemistry should be easier for every person to perform. It should be broadly applicable. It should be performed under mild reaction conditions. You have to avoid the high temperatures, special environment, inert, and toxic solvents. And it should have high yield and no chromatographic separation, meaning that the reaction should be clean. So all these kind of properties should be present in a reaction to be called as a click reaction. Okay. Regioselectivity and specificity should also be present, but essentially the inertial selectivity is not required, and it should have either no solvent use or some environmentally friendly solvents like water. So in other ways, chemistry should be like this. It should be fun, it should be easy to play with. And if we look at the spectrum of reactions at that time, we have actually very few reactions which fall under these categories. So first one is the opening of a strained three-membered system like epoxides or aziridine. So different type of molecules can react with it, and they normally give a clean reaction. Then we have cycloaddition reactions, and most of the students from chemistry background should know that what is cycloaddition reaction. And then some carbonyl chemistry like azide formation and heterocyclic formation. So these was the weaponry available to chemists to perform the click type chemistry at that time. Then comes this guy, Morton Meldon. He was actually performing a very straightforward, simple reaction in which he envisioned that, and this reaction is already by the way reported, that he was using a copper-based alkyne reagent and he wanted to react it with another reactive molecule to make this sort of linkage between them. The difference, what he was doing and what was already reported, it was that his molecule contained additional part in it. And this part was a kind of issue for them. So I have seen many times that if your supervisor gives you a project and asks you to do this, and you come up after several days and say that okay, it is not going to happen because you are not getting what you have planned. But if you are open to accept what nature is giving you, then sometimes you can win the Nobel Prize as well. Okay, so this guy, when he performed the simple reaction, this molecule reacted in the other way around. Even the reactive group was left intact. And this unexpected result behaves just like clicking the buckle together. So from here this term become famous, and interestingly Sharpless also reported the same reaction in the same year. So there was two evidences that alkyne and azide, these are two special functional groups which can snap into each other, which behave like buckling two molecules together to make a ring type structure which is very stable, and this stability is again due to the aromaticity of this compound. This reaction is now called as co-arc, which is copper based azide alkyne cycloaddition. Okay, so if I say co-arc reaction, that means the snapping together of alkyne and azide in the presence of copper. Okay, with the report of this reaction in 2001, you can see that the exponential growth of the publication which are using this reaction. So by 2011, every year around 2,000 publications were benefiting from this reaction, and that really indicate the ease of formation of the product using this chemistry. So anytime any building block which has a clickable alkyne and the clickable azide group, they can be joined together, they can mix together. And this type of reactions are ideal for the fragment phase drug discovery. Okay, so now I will give you few technological development which were actually developed by using this reaction, and these are actually very interesting ideas as well. So suppose you have different building blocks and you want to develop an inhibitor against enzyme for the drug discovery, then your enzyme will synthesize its own inhibitor on its surface. How this will happen? You mix this mixture of all alkyne containing with building block and azide containing building block and put your enzyme in that solution. then any building block which fit into the pockets of this enzyme, they will expose or bring the azide and alkyne together, and they will allow it to snap together within the enzymatic pocket. So all other molecules will not react with each other. Why? Because there is no copper around in this solution. So the only reaction will take place because of the constrained environment and the proximity of these molecules. Another technology which is developed by this click reaction was the Boncat, which is the bioorthogonal non-canonical amino acid tagging. So suppose you want to observe that what kind of proteins are formed when you expose cell with any material, any chemical. So what type of proteins will be formed? So for that, you prepare a amino acid surrogate which is tagged with azide. For example, methionine analog is this azidohomoalanine, and cell actually, the ribosomal machinery cannot differentiate between the natural amino acid and unnatural amino acid. So anytime the new protein is formed and this starting methionine amino acid is present in the culture medium, then it will be inserted as a first amino acid, and then the whole translation will take place, and then we will get the peptide. So all new peptides will have this azide as a tag. So if you treat the reaction mixture with the clickable alkyne group which contain fluorophore or any material which you can analyze later on, then it will trap or react with those peptides which are newly formed, which contain the azide functional group as the first starting material. Okay, another technology which was developed by click chemistry is the profiling of enzymatic status, or the presence or absence of any enzyme in the protein mixture. So if you have a cell lysate which have different type of protein, and you treat with substrate or inhibitor of that protein which are labeled with the azide or another clickable substrate is the diene, which undergo Diels as a reaction. So if you treat with that, then they will recognize their protein, they will bind tightly with them, and when you expose it with their clickable partner, then they will be either tagged or fluorescently labeled due to this reaction, and later on you can separate them or run them on gel electrophoresis, or you can perform the mass spectrometry or even in situ fluorescent by using confocal microscope. So these kind of things are possible with click chemistry. And the last technology is the metabolic labeling. That metabolic machinery can be used to label different type of biomolecules. For example, you can label DNA by using a nucleotide which is labeled with alkyne or azide. Proteins can be labeled with amino acid which have the tag. You can label the cell membrane by using a tagged fatty acid, or simply the sugar to label the glycone or the sugar coat of the cell. And after labeling, you expose them with a detection system which is again based on the azide alkyl chemistry, and you can determine the presence or absence of certain targeted proteins or molecules in the cell. And now from this part on, I will start the story of Caroline Bartuzzi, who was actually working in parallel to the development of click chemistry. She is a synthetic organic chemist by training, but she did postdoc in glycobiology and became interested to investigate the sugar coats on the cell surface. So each cell, or actually most of the cells, they have their surface which have a variety of sugars, like you see in this cartoon, that this is a very complex meshwork of sugar moiety. So each little box is representing some of the sugar, for example glucose, mannose, glucose, etc. And these sugars, they are continuously changing in response to different stimuli. For example, during development, growth development or disease development, this codes keep changing, and the sugars are either removed or new sugars are always added, and they can serve as a biomarkers for different type of diseases and the development stage. So she wanted to explore this area and develop some sort of bioimaging technique in the living cell. And existing technology at that time did not allow to explore this area unless you have to kill the cell and open the cell membrane and isolate all these components. So she needed something which can be used in the living cell to investigate the dynamic nature of this mechanism. Okay, so she was inspired by some work from the Werner Reuter, who actually published one paper in 1992 in which he has injected a rat with radio-labeled unnatural sugars, and then after some time the different organs were removed, and it was found that the sugars has been present in the cell surfaces of different organs like lungs, kidney, brain and liver. And from this finding, she thought that the metabolism in the cell, in the living organism at least, allow the utilization of unnatural biomolecules. So she thought that why not we explore the keto and azide chemistry. So ketone and hydrazide, they are very reactive. They show the properties like a click reaction, click chemistry. So she treated cell with unnatural sugar which have additional ketone moiety in it. And these cells were then exposed to the detector system, and it turned out that the cells are actually accepting these unnatural sugars, and they are putting in the sialic acid and other sugar moieties in the glycose. So the ketone hydrazide chemistry is not compatible with the experimental living organism, because ketone is present in different biomolecules, and there is issue of selectivity. So she actually thought that instead of keto hydrazide chemistry, it will be beneficial if we use the azide and phosphine chemistry. She has already worked in the azide carbohydrates, and there is a reaction already known which is called the Staudinger reaction, and azide can be reduced by phosphines, and this was the knowledge which existed at that time. And both azide and phosphine, they have been used in the cell, and the cells do not have any kind of toxicity and other issues, so she thought that why not we use this chemistry. And from there the bioorthogonal chemistry started, that to use a reaction which is developed in the lab in the cell. And for that she actually put forward some of the rules, that a bioorthogonal reaction should be one which does not exist in the cell, and it should not affect the cellular biochemical pathways, and it should be fast and complete, and it should not produce any waste or toxic material. And this was in other way two galaxies which are coexisting in the same environment. So here is the experiment in the cell level when she prepared this special sugar which is actually labeled with azide, and cell has metabolically placed it on the sugar coat, and when it was treated with the phosphine reagent, this is phosphorus here, so it will react with this azide to connect the detector system, which is biotic in this case. And that demonstrated that the azide and phosphine, they can be used in the click chemistry, at least in the cell level. But in animal levels, it need to be proved. So she used the azide sugar, injected mice for 7 days, isolated the splenocyte from mouth, and isolated the membrane from splenocytes, and treated it with the Staudinger ligation region, which contains a peptide which serves as a flag, and this peptide has the corresponding antibody which has a fluorescent dye labeled on it. So when they were mixed together, the membranes were detected by flow cytometry. So this indicates that this sugar, unnatural sugar, can be tolerated in the mouse, and it can be integrated in the glycobiome of the cell, and then they can be studied later on. Okay, so this reaction, which people started calling Bertuzzi-Staudinger ligation, this has one issue, that it required a longer time, like 8 or 9 hours incubation. And when this work was published, then click chemistry was already, the click co-arc reaction was already out, and she thought that we should try the Sharpless and Melder copper-based click reaction, because that's fast and that can basically remove the hurdles which she was facing in this Bertuzzi-Staudinger reaction. And this is her own quote, that she was actually teaching the first semester organic chemistry, and where she give lecture on the different strain systems and how different molecules they can be strained due to the presence of some chain bonds. So she finds that the strained alkyne in any range system would react faster, and maybe the issue of copper toxicity can be solved, which which is a catalyst in case of Sharpless-Mander reaction. So here is the original Sharpless-Mander reaction, which required copper, and here is the strained alkyne, which react with the azide in the same way to make same kind of ring. So this reaction is now called the click reaction without copper. Okay. So now once she figure out this reaction condition, she treated in the cell level. The cell were treated with this mannose and the compound which contained the azide, and the azides were coated on the surface glycome, and then they were treated with this strained alkyne, and the surface was labeled or reacted with this one. And this is shown with these pictures where these cells were illuminated with the green fluorescent dye, and here at 0 minute after the labeling, you can see that all green color is outside the cell, which indicate the presence of this sugar on the board. And once this after 15 minutes, so this sugar, they start internalizing and accumulating in the centrosome in the cell, which keep increasing by 30 minutes, and then it remains constant up to 1 minute. And then she said that how the glycones, they internalize and synthesize and are present on the surface of the cell. And lastly, she tested the same technology in the living organism. She used the zebrafish embryo, and the embryos were treated with this azide probe, and then exposed with the alkyne, strained alkyne detector system, and we can see the presence of all these sugars in different parts of the embryo. And the spatial or differential expression of different carbohydrates are shown with the different colors. For example, if you use a click reaction with the green dye, then that was done in first 60 minutes, and after 1 hour they were tagged again with the different dye, and it shows that the first dye, the green one, that start internalizing, and the red dye start appearing on the surface. So overall, she developed a biotechnical method in which you can explore the dynamic nature of sugars in the living cell. Okay, so in the end, these guys, all three of them were awarded the Nobel Prize based on the discovery of this click chemistry and bioorthogonal chemistry. And I thank you very much for your attention, and if you have any questions, please. Yes.
very live question. So the regular assembly of the, for example, large molecule like DNA from simple amino acids, why is it not a quick reaction? So आपको level loss और बात कर रहे थे, then you gave like 6 to 7 conditions. So कौन सी condition है जिसको this normal assembly equivalent?
Normal assembly of what?
So for example, the assembly of the DNA molecule or the RNA molecule from just amino acids, isn't that a quick reaction as well?
No, quick reaction is actually there is a reaction which is already going on in the cell, and if you want to investigate any process, you have to introduce some chemical in the cell, and most of the time the chemical which you introduce in the cell, they start reacting with the existing chemistry of the cell, which is called the inhibition. That is how the drugs actually interfere with the biological pathway. And now we need something which do not react with anything in the cell. And that is the uniqueness of that. For example, we can, make various drugs which keep on interacting with different pathways, but by orthogonal chemistry, we want a chemical or a reaction which remain hidden from the existing chemistry in the cell, and then they are analyzed based on what we want to do.
I think this question was a little bit different.
Yeah.
We wanted to know that